Imagine you're scrolling through a streaming app on a Friday night, and it recommends a deeply obscure, independent sci-fi thriller.
Oh, yeah, the ones you didn't even know existed.
Right. You've never heard of it. You definitely didn't search for it, but the algorithm just somehow knew your tastes perfectly.
Based on, what, 1,000 invisible data points?
Exactly. The pacing of the last 3 movies you watched, the directors you favor, even the specific color grading of the thumbnails you click. It saw a pattern you didn't even know you had.
It's spooky when it works that well.
It is. But now I want you to imagine if that exact same recommendation math could look at the molecular profile of your specific cancer cells and just, recommend the single drug that will stop them in their tracks.
Well, what's fascinating here is that this isn't just a hypothetical anymore. It's a profound shift in how we think about precision medicine.
Which is the mission of our deep dive today. We're unpacking how artificial intelligence is solving one of the most critical challenges in oncology.
Yeah, we're dissecting a breakthrough 2021 study by Corres and colleagues, published in Scientific Reports, and they tackle a massive hurdle, forcing a deep learning black box to actually show us its biological homework.
We want to know exactly how it predicts if a drug will work. And, for anyone following targeted therapies, you already know we're talking about kinase inhibitors.
Right, the absolute backbone of modern oncology. Kinase inhibitors are basically drugs designed to jam the biological switches, the kinases that get stuck in the on position, and fuel uncontrolled tumor growth.
Matching the right jammer to the right tumor is notoriously difficult. So to build their model, these researchers gathered a massive data set. They looked at 74 distinct kinase inhibitor drugs.
And they tested them against 922 different laboratory-grown cancer cell lines. It's a staggering amount of data.
Okay, so let's get into the building blocks here. To train an AI, you need a metric for success, right? Like a star rating for how well the drug worked.
Exactly. And in pharmacology, you're usually looking at the overall dose-response curve, which is often measured by AUC, or area under the curve.
Which is like looking at the entire battlefield over a long period of time to see how effective the weapon was.
That's a good way to put it.
Yeah.
But for this specific AI model, the researchers found that AUC was actually too noisy. The metric that proved vastly easier for the AI to learn was the IC50.
The half maximal inhibitory concentration. Okay, I love a good visual memory trick for this.
Oh, go for it.
Picture an army of exactly 100 cancer cells sitting in a petri dish. The IC50 is the precise concentration of a drug, literally down to the micromolar drop needed to kill exactly 50 of those cells.
Right, so logically, a highly potent drug is going to have a very low IC50 number.
Because it takes barely any of the compound to wipe out half the invading force, so low IC50 equals a great drug match.
Exactly. That's the target the AI is trying to predict. But to get there, the inputs, the data they actually fed into the system are incredibly dense.
Let's break those inputs down, starting with the cell lines. They fed the AI 241 distinct features for every single one of those 922 cell lines.
Yeah, it's a high-resolution snapshot. It includes the mRNA expression of 202 different genes.
And for anyone who needs a quick refresher, mRNA expression is basically a measure of how loudly a specific gene is shouting its instructions to the cell, right?
Oh, precisely. They also included 21 binary mutations.
Yep.
So is this critical gene mutated, yes or no? And 18 tissue types.
Like, did the cancer originate in the lung, the pancreas, the skin, that sort of thing? So we have this highly detailed map of the cancer's environment.
But the drug data is where this gets really interesting. They didn't just tell the AI what the drug was supposed to target.
Right. And this is where I actually found myself pushing back when I read the paper. They fed the system the drug's binding strength across a massive panel of 294 different protein kinases.
They mapped its whole interaction profile, yeah.
But wait, let's unpack this. If a pharmaceutical chemist designs a highly targeted drug specifically to inhibit, a mutated BRA kinase, why are we cluttering the AI's input with 294 other random kinases?
It seems counterintuitive, doesn't it?
Yeah, shouldn't we just focus the AI on the intended target to reduce the noise?
It's a totally natural assumption, and the researchers actually tested that exact hypothesis. They ran what's called an ablation study.
Where you selectively remove parts of the data to see how the AI reacts.
Right. They stripped away the full 294 kinase profile. They restricted the AI, letting it only see the known primary targets of those 74 drugs.
And what happened?
When they forced the AI to focus only on what the drug was designed to do, the model's accuracy dropped by over 11%.
Wow. And in the world of predictive bioinformatics, an 11% drop is basically falling off a cliff.
It fundamentally ruins the prediction. What this proves is that the off-target interactions are secretly doing a massive amount of the heavy lifting.
Because drugs are inherently messy molecules, right? They aren't perfect little heat-seeking missiles.
Exactly. A therapy might be engineered to hit target A, but as it floods the cellular system, it accidentally bumps into and inhibits targets B, C, and D.
And historically, pharma companies view those off-target effects as a nuisance, like they're the primary cause of toxic side effects.
But this AI flipped that paradigm completely. It recognized that the collateral damage a drug causes is highly predictive of its success in actually killing the cell.
Okay, that is a huge perspective shift. So let's do a quick 5-minute recap and bullet summary for you listening. Bullet 1.
We are trying to match 74 kinase inhibitor drugs to 922 cancer cell lines.
Right. Bullet 2. The ultimate goal is predicting the IC50, which is exactly how much drug is needed to kill half the cancer.
And bullet 3. A drug's accidental off-target effects are actually highly predictive of its overall success.
Which brings us to an audience question we got that fits perfectly here. If you were designing a new therapy, would you focus purely on its main intended target or actively design for its off-target collateral damage?
Well, based on this paper, you absolutely have to map and leverage its messy, real-world footprint. You can't just obsess over the primary target anymore.
But that raises a massive technical problem. How do you actually feed all that messy data, the 294 kinases and the 241 cell features, into an algorithm without it just memorizing the noise?
That brings us to the engine itself. The researchers built a bespoke architecture named DEERS.
DEERS. That stands for Drug Efficacy Estimation Recommender System.
You got it. And at its core, DEERS builds on matrix factorization, which is a classic recommender system technique.
Like how Netflix matches users to movies, but here we're matching cell lines to drugs.
Exactly. But basic matrix factorization wasn't enough. Biological systems are infinitely more complex than movie preferences, so they upgraded it using deep learning.
Specifically, they used autoencoders, right?
Yes, autoencoders were used to process the input data.
Okay, let's visualize how an autoencoder works because it's brilliant. Think of it like taking a massive 241-page technical novel, which represents all the complex genetic data of the cancer cell, and handing it to an editor.
A very ruthless editor.
Right. You force that editor to summarize the entire plot using exactly 10 words. That 10-word summary is what they call the bottleneck or the hidden dimensions.
And then you hand those 10 words to a second writer and tell them to reconstruct the original 241-page novel perfectly.
Just from those 10 words. And if they can do it, it proves that the 10-word bottleneck captured the absolute, undeniable essence of the original data.
It's a perfect analogy. And DEERS uses two of these editors simultaneously. One autoencoder compresses the cell line's 241 features down to 10 hidden dimensions.
And a separate autoencoder takes the drug's 294 kinase interactions and compresses them down to their own 10 hidden dimensions.
Right. But the real ingenuity, the mathematical trick that makes DEERS so stable, is found in how they tweaked the cost function during this compression phase.
Now, the cost function is essentially the grading rubric for the AI. It tells the AI what a good job looks like. How did they change it?
They added a specific penalty enforcing independence. They programmed the cost function to actively punish the network if any of the 10 hidden dimensions overlapped in meaning.
Oh, that's clever.
Yeah, by minimizing the covariance between the dimensions, they mathematically forced each of those 10 numbers to represent something completely unique.
It's like having 10 different detectives working a crime scene. If they all walk into the kitchen and investigate the exact same footprint, you've wasted your resources.
Right, you learn nothing new.
But by forcing independence, you mandate that Detective 1 looks exclusively at the DNA evidence, Detective 2 looks only at the financial records, Detective 3 interviews the witnesses, and so on.
You force a much wider, non-redundant understanding of the problem. That's exactly it.
So once you have these 10 perfectly independent dimensions for the drug and 10 for the cell line, what happens?
You bring them together. Those 20 dimensions total are fed into a final feed-forward neural network.
And this network is pretty robust. The paper mentions it incorporates hidden layers with 512 and 256 neurons, using ReLU activation functions to handle nonlinear patterns.
And crucially, a 0.5 dropout rate to aggressively prevent overfitting.
Dropout is literally shutting off half the neurons randomly during training, right? It ensures the network doesn't just rely on one superstar neuron to make the prediction.
Exactly. It forces the entire network to learn the underlying patterns, and the results speak for themselves. DEERS achieved a massive 0.86 correlation score on entirely unseen cell lines.
That is huge. It matched the predictive power of heavy-hitting models like XGBoost, and it completely blew standard matrix factorization out of the water.
It did. But, achieving a high correlation score brings us to the central tension of deep learning in medicine.
The black box problem.
Yes. We have this phenomenal 0.86 accuracy, but neural networks are famously opaque. In a clinical setting, a physician cannot responsibly prescribe a grueling chemotherapy regimen simply because a black box outputted a high probability score.
There has to be a why. The algorithm absolutely must be interpretable.
Right.
Let's do our 10-minute recap and bullet summary before we solve that black box puzzle. Bullet 1. DEERS acts like a digital funnel, compressing huge biological data sets into 10 crucial dimensions.
Bullet 2. A secondary neural network mixes those compressed dimensions to predict the exact drug dosage needed.
And bullet 3. Mathematically forcing the 10 dimensions to be independent makes the AI vastly more accurate.
Which perfectly lines up with an audience question we received.
Yeah.
Why do you think an AI needs to compress data to understand it, rather than just looking at all the raw information at once?
Well, it's about finding the signal in the noise. If it looks at everything, it just memorizes the noise. Compression forces it to find the actual structural rules of the biology.
Exactly. And that brings us to the pivot point of the entire study. How do you force DEERS, which is dealing with highly abstract mathematical dimensions, to explain its biological reasoning?
Right, in a language human oncologists can actually understand.
They executed a highly novel two-step interpretability approach. They focused entirely on those 10 hidden dimensions that compressed the cell line data.
Okay, step 1.
Step 1 utilized integrating gradients. This is a sophisticated mathematical way of tracing the prediction backwards.
Like reverse engineering the math.
Yeah. It allowed the researchers to say exactly which of the original 241 input features, for instance, a specific mutation in a gene was strongly activating which of the 10 hidden dimensions.
So they successfully mapped the known inputs to the hidden math. That's cool, but step 2 is where the researchers did something I found completely audacious.
Oh, the genome-wide search.
Yes. Instead of restricting their analysis to the 241 features DEERS was originally trained on, they took those 10 hidden dimensions and checked them against everything.
They correlated them against the expression of all 17,419 genes in the entire human genome across those cell lines.
Let me make sure the magnitude of this lands for you listening. The AI only ever saw a curated list of 241 features during its training phase.
Right, a tiny fraction of the genome.
But to test if the AI had actually learned fundamental biology, they tested the AI's internal math against over 17,000 genes it had never even been exposed to.
They were actively searching for biological truth hidden in the math. They took these genome-wide correlations and ran them through gene set enrichment analysis, or GSEA.
GSEA is essentially a massive biological dictionary, right? It groups huge sets of genes into known biological processes.
Exactly. We call them gene ontology, or GEGO terms.
And the results they got back are just stunning. They discovered 67 distinct, recognized biological processes that perfectly map to these 10 hidden dimensions.
It proved the compression wasn't just a statistical parlor trick. The math actually mirrored reality.
Give us some examples, because this blew my mind.
So, dimension 2 perfectly aligned with the genes responsible for DNA replication and the cell cycle.
Wow.
Dimension 4 was entirely driven by protein metabolism, and dimension 8 unexpectedly captured nervous system development.
And you have to keep in mind, the AI has no concept of what DNA replication or protein metabolism is. It doesn't read textbooks.
No, it was simply minimizing a cost function. It was just finding non-redundant mathematical patterns in a data set.
But by correlating those mathematical patterns against the entire genome, they proved the AI had independently reverse-engineered actual foundational cellular biology.
It is profound.
It really is. The AI looked at a spreadsheet of raw numbers and independently discovered the biological themes that govern life and cell death.
Which is the true leading edge of computational biology today.
Time for our 15-minute recap. Bullet 1. Researchers crack the AI black box by mapping its hidden math back to real human biology.
Bullet 2. By testing the AI against 17,000 plus genes outside its training data, they proved it learned genuine biological truth.
And bullet 3. Each of the AI's 10 hidden dimensions acts as a distinct biological theme, like DNA replication or protein metabolism.
Which leads to a great audience question. If an AI can independently discover the concept of DNA replication just by looking at math, what other biological processes might it see that humans have completely missed?
That is the million-dollar question. If it can find what we know, it can definitely find what we don't know yet.
Absolutely. But to truly understand the value of DEERS, we have to pull this out of the theoretical math and apply it to clinical reality.
Yes, what does this all mean for an actual patient? Let's look at some real-world case studies from the paper, starting with Dabrafenib.
So Dabrafenib is an FDA-approved targeted therapy heavily utilized for metastatic melanoma, which is an aggressive form of skin cancer.
And its primary textbook mechanism of action is targeting a specific mutation in the BRAF gene, right?
Right. And when researchers asked DEERS to explain its predictions for Dabrafenib, the model confirmed that textbook knowledge. It correctly identified that the presence of the BRAF mutation and the skin tissue label were highly important.
Good, so it knows the basics, but it didn't stop there.
No, it didn't. The model revealed that the drug's success in actually killing the cell was heavily, almost exclusively, reliant on hidden dimension 4.
And from our genome-wide GSEA mapping, we know dimension 4 is the biological theme of protein metabolism.
Exactly. And digging deeper into dimension 4, it revealed that specific factors like neutrophil-mediated immunity and NF-kappa B signaling fundamentally dictate if Dabrafenib will work.
Think about what that means clinically. It shows that it's not enough to just have a drug that successfully hits the BRAF mutation.
Right. If the cell's broader immune signaling environment isn't properly aligned, the cell might just survive anyway.
The environment dictates the efficacy just as much as the primary target. It adds an incredible layer of nuance. The drug might do its job chemically, but fail biologically.
Let's contrast that with two other drugs highlighted in the study, PHA 793887 and XMD 1499.
Okay, these are two completely distinct compounds. PHA is designed to target CDK enzymes, while XMD targets ALK and CDK7.
Right, so in a clinical trial setting, a doctor views these as distinct therapies attacking distinct biological pathways.
Yet, when DEERS analyzed them through its compressed dimensions, it revealed a startling similarity.
It really did. Despite their different primary chemical targets, DEERS found they share the exact same top 3 cell line dependencies to be effective.
For either of these completely different drugs to successfully kill a cancer cell, that cell must express the BTK, PIM2, and TEC genes.
Furthermore, both of these drugs rely heavily on hidden dimension 3, which the genome map tells us is the dimension governing DNA and RNA metabolism.
Okay, so why does it matter clinically that two chemically different drugs share the same biological dependencies?
Because it completely rewires clinical trial design and drug repurposing. If a patient's tumor doesn't express those specific bystander genes, BTK, PIM2, and TEC, then neither drug is going to work.
Regardless of what the label on the bottle says they target.
Exactly. By understanding these shared dependencies, we move away from classifying drugs simply by their chemical design. We start classifying them by their actual biological footprint.
The researchers actually took this concept to its logical conclusion, didn't they? They didn't just analyze a few isolated drugs.
No, they clustered all 74 drugs against the 67 biological processes they discovered.
They essentially created a master map, like a periodic table of cancer biology.
And if we connect this to the bigger picture, the patterns that emerged from that master map are systemic insights that humans simply couldn't spot by staring at spreadsheets.
Like with the PI3K-mTOR pathway drugs.
Yes, that is a critical pathway for cellular growth. Humans naturally group these drugs together based on their structural design. But DEERS revealed something deeper.
It showed that all the drugs targeting that specific pathway share a unique underlying reliance on cell junction assembly and epidermis development.
Insights we couldn't easily spot alone.
So, mini summary here. By applying DEERS, we aren't just getting an automated system that spits out a dosage recommendation.
No, we uncover hidden similarities between distinct drugs and gain a master blueprint of how therapies manipulate biological pathways.
Which perfectly tees up our final audience question. Could a master map like this be used to take a drug that failed its clinical trials and perfectly match it to a different type of cancer it was never originally intended for?
Absolutely. Because DEERS operates on fundamental biology rather than human-assigned labels, the potential for rescuing failed drugs is massive.
It completely changes the scale of what's possible. And that brings us to the core reason why you, listening to this, need to care about DEERS. It isn't just because it's cool math.
It's deeply personal.
It is. Because when you, or a friend, or a family member face a cancer diagnosis today, the standard of care is far too often a grueling cycle of trial and error.
You endure a chemotherapy regimen, you suffer the severe toxicities, and then you wait months for a scan.
Just to see if the tumor actually shrank. And if it didn't, the doctors pivot and you try another drug. It is physically devastating, and it costs patients time they often do not have.
But an interpretable AI like DEERS offers a glimpse into a future where oncology transforms from an educated guessing game into a precise, targeted, and fully explained science.
By mapping the exact molecular vulnerabilities of a patient's specific tumor to the messy real-world profile of a drug, we can skip the trial and error phase entirely.
That is the ultimate promise of precision medicine. However, I want to leave you with a final puzzle to mull over, because the work is far from done.
What's the catch?
Well, DEERS achieved this incredible accuracy by analyzing static cancer cell lines cultivated in a laboratory dish. Those cells are snapshots frozen in time.
Right. But a tumor inside a human body is an intensely dynamic ecosystem.
Exactly. It evolves, it mutates to survive the drugs you throw at it, and it's constantly fighting a chaotic war with your immune system.
So the tumor you biopsy on day 1 is genetically distinct from the tumor you are fighting on day 30.
So here is the provocative final thought. If this AI can so perfectly predict the behavior of static cells, how will recommender systems need to adapt when the user, the cancer, actively mutates to resist the recommendation?
How do you map the biology of a moving target? That is the puzzle waiting for the next generation of deep learning models. But for now, this deep dive proves that the days of throwing darts in the dark at cancer are numbered. Thanks for joining us, and we'll catch you on the next one.